My group studied the effect of varying starvation and log phases on TOR1 expression in tetrahymena thermophila. The overall goal of this experiment was to potentially design an assay system that will allow us to screen for new autophagy drugs. In order to design a potential assay system, we needed to find a gene whose expression is altered during autophagy in tetrahymena thermophila. In this experiment, we tested our chosen gene, TOR1, for changes in expression during starvation phases and log phases. Today's research is trying to find treatments and therapies for diseases like Crohn's disease and Parkinson's disease that are brought on by the malfunctioning of autophagy in a person's cells. Our potential assay system with TOR1 would allow researchers to use the gene as a drug screen to test compounds that affect autophagy. We chose TOR1 because it is a protein kinase that regulates cell growth and, when inhibited, induces autophagy. Since it plays such a key role in autophagy, we wanted to see how its expression was altered when placed in stress-induced conditions. The overall design of this experiment was to choose a gene that is related to autophagy, design primers for the chosen gene, and use PCR to see if the chosen gene's expression changes during starvation phases and log phases. For this experiment, our hypothesis was TOR1 expression decreases during the starvation phases of 12 hours and 24 hours and increases in the log phases. For our methods, first we grew our tetrahymena thermophila that had the DNA we are going to use in this experiment. Second, we designed our primers for the TOR1 gDNA and cDNA. Third, the gDNA was taken from the tetrahymena thermophila and was then used to synthesize the cDNA. Fourth, we prepared to run our PCRs by finding our concentration, sample sizes, and controls. The 1.5% agarose gel and the concentration of the working stock solution of DNA was determined by how many base pairs we were expecting, and the sample sizes were determined if we wanted to duplicate or triplicate what we were testing in case there were errors. Fifth, we ran a PCR to test our primers. Sixth, we tested TOR1 with our design primers for changes in expression during our starvation times of 12 hours and 24 hours. Once our PCR was finished, we analyzed the gel for changes in expression. What is unique about our experiment is that the expression of TOR1 should decrease drastically during starvation. Since TOR1 thrives in nutrient-rich conditions and is inhibited by stress-induced conditions, there should be a significant decrease of expression during our starvation phases shown in our gel. This decrease in expression shows that TOR1 is important in autophagy since the inhibition of TOR1 drives ATG13 to bind to ATG1 and start the process of autophagy. Our data is shown below in figures 2 and 3. In figure 2, the gel above shows our primers that we designed for the TOR1 gene. Due to the size of the band, we know that our primer worked. We ran our positive control, negative control, and gDNA with our design primers, all as duplicates. In figure 3, the gel above shows our positive and negative controls, log phase cDNA, and our gDNA at 2 starvation times. Both gDNA and cDNA were run with the primer we created. Starvation time 1 is 12 hours, and starvation time 2 is 24 hours. As for our results, in figure 2, we tested our design primer, which worked based on the band size. We expected a band of around 660 base pairs. According to our ladder marker, we are at 700 base pairs. With this confirmation, we were then able to run our starvation phases and log phases using the primer and gDNA as our positive control. The gel in figure 3 allowed us to test our hypothesis. Lanes 3 and 4 have no band because there is no DNA and is our negative control. Lanes 4 and 5 contain our positive control, which matches up with the band size in figure 2. Lanes 6 through 9 contain the log phase cDNA in our primer. Lanes 6 and 7 show normal amplification, which is consistent with our hypothesis. Lanes 8 and 9 show a decrease in amplification, which is inconsistent with our hypothesis, and could be due to the increase of 2 microliters of cDNA as shown in Figure 4. Lanes 12 through 15 contain the S1G DNA, which shows a slight decrease in amplification and is consistent with our hypothesis. Lanes 16 through 19 contain the S2G DNA, which shows a decrease in amplification and is consistent with our hypothesis. As for our conclusion, according to our JAL in Figure 3, the expression of TOR1 decreases when in starvation phases and decreases when in log phases. According to what we originally hypothesized, the expression of TOR1 in starvation phases decreasing is consistent, while the expression of TOR1 in log phases decreasing is not. When a cell is stressed and enters into starvation, our findings show that the TOR1 expression decreases. This inhibition induces autophagy by forcing ATG13 to bind to HEG1, and its expression is altered and lowered during starvation. This finding could help design a drug assay system that would allow researchers to use the gene as a drug to test compounds that affect autophagy. This could lead to treatments and therapies for diseases that are created due to the inhibition of autophagy in cells, like Crohn's disease and Parkinson's disease. Overall, our experiment went very well, but a logistical challenge that affects the entire experiment design could be not enough pipetting practice. Pipetting is extremely important in creating correct concentrations for both the PCR components and in placing them in the wells of the gel. As for future directions, in the future experiments, we would better analyze the variety of starvation times to better understand when it is least expressed. From our findings, we saw that TOR1 was least expressed given more time in starvation phases, so we should test starvation phases past 24 hours. Figure 5 shows the gene expression charts with starvation phases in blue.